Hi children, today we are going to learn about molecular basis of inheritance, one of the difficult lesson to understand. So here in this lesson, I am going to give you the MCQs and also the correct answer as well as you can see the wrong answers explanation also. That means in this lesson we are going to learn about DNA structure and replication, RNA types and transcription, protein synthesis, translation, transcription also, lack of operon and gene regulation. The molecular basis of inheritance explores DNA and RNA genetic material. Their structure, replication, transcription, as I said, we are going to learn. So here you can see the slide. Removal of introns in a transcription unit is, you can see the answer here, splicing. And why this is correct in the sense, process in which introns are removed from the primary RNA transcript and exons are joined. So this is the correct answer. You can see in the picture also. Capping is the addition of 5' methylguanosine and tailing is the addition of poly A tail at the 3' end. Transformation refers to the uptake of foreign DNA by bacteria and is unrelated to processing, RNA processing. The next question, genetic information along with the answer I gave you here, transferred from the nucleus to the cytoplasm via RNA, specifically this is mRNA. Why the other three are wrong? RNA is the correct answer. During the transcription, DNA in the nucleus is copied into messenger RNA, that is mRNA, and the mRNA leaves the nucleus through nuclear pores and carries the genetic information to the ribosomes in the cytoplasm, whereas proteins are synthesized. Remember, the proteins are synthesized in the cytoplasm. Why the other three options are wrong? So one is anticodon. It is a part of tRNA that pairs with the codons during translation. It does not carry genetic information from the nucleus. And why DNA is not the correct answer? Because DNA remains inside the nucleus in eukaryotic cells. And lysosomes, of course, they are organelles involved in the intracellular digestion and have no role in transferring genetic information. Along with the one question, you are going to learn the 4 questions of this slide, every slide. Next one, the commonly used vectors for human genome sequencing are, answer is BAC and YAC. I think you know that bacterial artificial chromosome and yeast artificial chromosome can carry large DNA fragments. They were widely used in human genome project for cloning and sequencing large pieces of human DNA. TA is the cloning vector, are mainly used for PCR polymerase chain reaction product cloning. Expression vectors are used for the protein production, and tDNA is used in plants genetic engineering. Next question, the association of histone H1 with a nucleosome indicates the DNA is considered in the chromatin fibers because histone H1 is called linker histone. It binds to the linker DNA between nucleosomes. This helps in folding the beads on string. This picture is there in your textbook, children. Structure into 30 nm chromatin fibers resulting in DNA condensation. It is not directly involved in transcription or DNA replication. There is another core question. Here you can see core histones. H2A, H2B, H3, H4. In the picture it is given all this. Form the nucleosome core. Linker histone H1 helps in chromatin condensation with the 30 nm fiber. Next question. DNA fragments are, of course, you know this, negatively charged because DNA contains a phosphate backbone, and this Phosphate groups carry negative charge, making DNA molecule negatively charged. This property allows DNA fragments to move towards the positive electrode anode during gel electrophoresis. When you learn this, you will understand this. It's a very important question. Once again, we can see here that if hysteresis coli grown in 15 N medium for several generations, and after that it is allowed to grow in 14N, that means 15N is a heavier nitrogen and 14N is the lighter one, medium for the one generation. Which of the following would be correct with regard to the first generation bacteria in 14N medium? The answer is B, that is both strands of DNA would have different density but resemble parent DNA. This is because on semi-conservative DNA replication, this was given by Michelson-Stale experiment. It is there nicely clearly in the textbook. After several generations, in N15, the parental DNA is having N15 and N15. But after transferring the bacteria to N14 for one generation, you can see here N1515 and N1514 plus N1514. And you can see, so each daughter DNA molecule has one old 15N strand, that is a parental strand, and one 14 strand newly synthesized strand. Therefore, the two strands have different densities and the DNA resembles the parent DNA because it retains one parental strand. The next question is, which of the following pairs of codons including all three chains termination codons? You can see here the answer is B, UAG, UGA and UAA. UAA is ochre, UAG is amber and UGA is opal or nonsense codons which do not code for any of the amino acids. So these therefore cause the termination of translation. Next question, consider the following. The Lac-Operon model, here the diagram is given here, has three elements, regulator, operator and structural gene. I think you know this. And the prokaryotes DNA has both exons and introns. And RNA polymerase binds to promoters and initiates transcription. And the regulation of Lac-Operon in E. coli occurs at epigenetic level. The answer is A, 1 and 3 are correct. Eukaryotes genome has unique feature of exon introns organization of protein coding genes representing coding sequence and intervening sequence that represents the functionality of RNA part inside the genome. The regulation of gene expression in prokaryotic cells occur at the transcription level. The reason for the anti-parallel strand of DNA is, we know that anti-parallel to each other, 5' and 3'. Here you can see hydrogen bond, ionic bond, phosphodiester bond and disulfide bond. Phosphodiester bond is the correct answer because the DNA strands are anti-parallel because of sugar-phosphate backbone has the directionality 5', 3' determined by the phosphodiester bond. One strand runs 5', 3', the other 3', 5'. We know this. Hydrogen bonds hold complementary bases together, but they do not determine the antiparallel orientation. So the correct answer is phosphodiester bond. Next one. In a transcription unit, the promoter is situated towards 5' end of the structural gene is the correct answer because the promoter is located upstream. See the picture of the structural gene. It polymerase binds to initiate transcription. The primer in DNA replication is answer is small ribonucleic polymer because a primer is a short RNA ribonucleotide strand. This is a very, very important answer. Synthesized by the enzyme primase. DNA polymerase cannot start DNA synthesis on its own. It requires a free 3' OH end, which is provided by the RNA primer. Therefore, the primer is a small nucleotide polymer, not DNA. Why the other options are incorrect? Because helix destabilizing protein. Remember, helix destabilizing protein, it keeps the DNA strands separating during replication. It is not the primer, remember. Small deoxyribonucleotide polymer primers are made of RNA, not DNA. So, enzyme joining nucleotides of new strands, this refer to DNA polymerase, not the primer. Next question. With regards to the mature RNA in eukaryotes, which of the following is true? Exons appear, but introns do not appear in the mature mRNA strand. The primary RNA transcript, hnRNA, contains both exons and exons coding regions, introns non-coding regions. During RNA splicing, introns are removed and exons are joined together. Therefore, mature mRNA contains only exons. These are reasoning questions, all these are. In E. coli bacteria are grown for several generations in the medium containing NH4Cl15 as the only nitrogen source. The bacteria are then transferred to the medium containing NH4Cl lighter 14. After one generation, the DNA molecules will be the C answer. All are hybrids and one parental strand and one is synthesized strand. You can see that is N15 and one N14. DNA replication in is semi-conservative. Each parental strand acts as a template for a new strand. Initially, this is the parental strand. After transfer N14 medium, each old N15 strand pairs with a new N14 strand. Therefore, after one generation, all DNA is synthesized and N15, N14. This is also a very important neat question. Which of the following is true with respect to AUG? The answer is D, correct. It codes for methionine only. It is an initiation codon. It codes for methionine in both prokaryotes and eukaryotes. AUG codes for methionine in eukaryotes. It serves as an initiation, that is the start codon for protein synthesis in prokaryotes. AUG is also the start codon, but it specifies formyl methionine FMET, a modified form of methionine. In many NEET questions, this is accepted that AUG codes for methionine in both pro and U. Hence, all the statements are correct. Next question. The human chromosomes with the highest and least number of genes are respectively, clearly answer is given here, chromosome 1 and Y. Because chromosome 1 has the highest number of genes, around 2000 genes, and Y chromosome has fewest genes, around 50 to 70 functional genes. Therefore, the correct answer is chromosome 1 and Y. The equivalent of a structural gene is cistron. You can see here clearly. Cistron structural gene. The cistron is a segment of DNA that codes for a single polypeptide or functional RNA. It is another term for the structural gene. Operon is a group of genes regulated together. Mutant is the smallest unit of mutation. And this is here your senior faculty member of biology, and I'm having 33 years of experience, children. I've been working for 33 years for NEET as well as senior secondary biology. Mrs. Roja Rajni Geddam, and I hope you people will like, share and subscribe. Thank you very much and all the best.